Then after that, I also talked about, it's not really a new model, but it's actually a modification of the HTLV1 model, because we're trying to prove a point that once, if the response function used in the model has a particular geometry or has a particular profile, then we are going to have transient oscillation. So that's why I'll be presenting a model, a modified HTLV1 model with a new CTL response function. First, about the biology evidence for transient oscillation. So HCMV is an acronym for human cytomegalovirus, and it's actually an infection that is often observed in bone marrow transplant recipients. So this data that I'm presenting to you is the viral load of CMV cytomegalovirus. And if you look at, so just before, I think this is about 90 days after transplantation. So between 0 and 90, we can see that there is oscillation in the viral load of the particular patient that is being considered here. So, and after 90 days, the oscillation actually disappears. So in this case, we have, so this is different from sustained oscillation, and it's also different from decay, because the amplitude of the oscillation is not continuously decreasing. So we have, in a finite period of time, there is oscillation, and at the end of the period, the oscillation disappears. So in the second example, this was actually a study to see the effect of drugs ZDV and LNV. So the study was actually to see the effect, to test the effect of the drug on the HTLV1 DNA copy numbers. So the point of interest to us here is the point where the drug was switched. So if you follow this line up to this point, so the curve that we have, the low curve is actually that of HTLV1 DNA copy number. So when the drug was switched, you can actually see oscillation. And this is, again, this is not sustained oscillation, and it is not also decayed oscillation. But at the end of so this is 224 weeks after the treatment, we can see that the oscillation actually disappears. So again, the question is, what is actually the type of mechanism that produces such oscillation? The third example that I'll be talking about is the one, it was actually an experiment where colonies were actually infected with ERA-B, which is an equine infectious anemia virus. So the horse was infected, experimentally infected with the virus. And as you can see from here, so the black diamond that we have there represents the viral load. So these are just like HIV for horses. So we can see that there is oscillation. So if you follow it from this point, so there is oscillation. And about 400 days after infection, the oscillation also disappears. So again, in summary, in fact, we're talking of oscillation within a finite period of time, and at the end of the time, the oscillation disappears. So, which is, at least it has not been studied in the literature. And so the first, I'll be talking about the first model that actually tries to study or explain, give a mathematical reason for transient oscillations. So, but before then, because we have to take into consideration CTL response function. So in the literature or in biology or immunology, the general shape of CTL antiviral response is what we have here. So this red curve represents the viral load level, and the blue one is the CTL response curve. So the interesting thing here is, if we do this actually theoretical anyway, because in reality the viral load is never clear. So if you look at this, there are four phases here, but two interesting phases are the induction phase and the AICD phase, activation induced cell death phase. So we can see that it is in these two phases that the viral load gets cleared. So it is then looking at this curve, one should think that if we were going to get a model CTL response, we should use a function that actually mimics or behaves like this general profile. So the first model that actually explains transient oscillations is that of HTLV, used by Dr. Lee and his former student, John Lam. So this is a 3-compartment model. So the first compartment represents the number of LD64 cells. I'm not really going to explain the biology and all that, because the point is actually to show that transient oscillations can be observed. So the first compartment, like I said, the number of LD64 T cells, and the second one represents the infected CD4 cells or provider CD4 cells, while the third compartment represents CTL cells. So that's the compartment for the immune response. Actually, there are 2 types of immune response to HTLV. So we have the antibody immune response and the CTL response. But for the antibody response, we're using these sigma, which represents the fraction of the cells of the infected or provider cells that survives antibody immune response. And for the CTL response, we're using a sigmoidal CTL response function. And so these, there were actually previous models for HTLV1, but the only one where transient oscillation is observed is that use this function, this sigmoid function, to model the CTL response. And that is the function that we have here. So I actually chose N to be 2 and A to be 2. So that will produce this curve. So these are other functions or CTL response functions that have been used in literature, but the only one that actually, at least up to now, that produced transient oscillations is the one that used CTL, the sigmoidal or logistic response function. So when the model was analyzed, there were actually 4 equilibria. So these are usually following the bifurcation study or bifurcation analysis of the model. So there were 4 equilibria. So this P0 represents the 6-free equilibrium, and this is another branch of equilibria that represents the carrier. So in this case, there is no CTL response. So it's just a carrier equilibrium. And the other equilibria that are of interest to us are these 2 branches. So if you look at the 4th equilibrium, the 4th branch here, so there is a switch of stability, so from unstable to stable. And this type of bifurcation is actually off bifurcation. And the explanation, so from what we have here, so it is this to reproduce the transient oscillations. So we actually chose the initial conditions that are very close to this stable manifold of this branch of equilibrium. And with those initial conditions, we're actually able to produce these transient oscillations. So what we're actually trying to do now is that we